Hello everyone, good evening to all of you. So we are starting a special lecture series designed for students who are preparing for NEET examinations, and this is a lecture series on biology. Similarly, we will have lecture series for chemistry as well as for physics. And based on your feedback after this class and your recommendations and suggestions, then we will take up more chapters which are actually difficult for all of you, so that we can discuss those, and the questions that are most likely come in the exam, we will also take up those questions as well. So let us wait for few minutes till everyone joins, and then we'll start. So we'll wait for 2-3 minutes at the max, and then we'll start after that. So we have Hardik and Dipali who has joined us online. So Hardik and Dipali, please feel free to ask and post your questions, okay, at any given point of time. In case you will have any clarifications or any doubts, I request that you post your questions as soon as the doubt comes in your mind, okay. Don't keep it till the last minute, okay. So let the session be as much as interactive as possible. Well, let's begin, okay. So before discussing about the topic for the today's discussion, which is bacteriophage, Lytic and Lysogenic cycle, let us discuss what are bacteriophages, okay. So bacteriophages are a group of viruses that infect the bacteria. So that is why they are called as bacteriophages. So this is the content that we are going to, or the discussion that we are going to follow today. So we'll discuss what are viruses, their general characteristic, what are bacteriophages, the structure of bacteriophage, and will specifically give maximum emphasis on Lytic and Lysogenic cycle, because most of the questions in the examinations usually come from these two parts, okay. And then we'll talk about the relevance of this particular chapter, and then we'll take up some questions, okay, which usually comes in the NEET examination, okay. So let's begin, okay. So this is usually the characteristic feature of viruses. So viruses are acellular organisms. So when I say acellular, means they do not have a cell based organization. As we know that cells are the structural and functional unit of every living organism. And if viruses do not have a cell based structure, will they be living? Oh, well, that is a very interesting question. They are called as non-living outside the host cell, but they behave as living entity inside the host cell, okay. So they are acellular in nature, they do not have any cellular structure, and as they are acellular, therefore they are obligate parasite. Obligate parasite means they are completely dependent on the host for its survival. They cannot live on its own, because a cell structure is essential for metabolic processes. As they do not have a cell based structure, they cannot do metabolism. As metabolism cannot be done, so therefore they have to depend on other organisms for its survival. So therefore they are called as obligate parasites. Obligate means they are completely dependent on the host. And as I said earlier, they are in, these viruses are non-living outside the host cell. They are just as an inert particle. But as soon as they enter into the host cell, they take control of the host cell machinery and try to become active, okay. So outside the host cell, they are not called as viruses, they are called as virions. So this is a very, very important thing that you have to remember, that outside the host cell, they are not called as viruses, they are called as virions, okay. So this is the most important part that you have to remember, okay. The structure of virus, as you can see here, is a very simple structure. They have a nucleic acid core, as you can see here. The nucleic acid can be either DNA or RNA. They don't have both. Okay, so they have either the DNA or the RNA as their genetic material, and simply a coat which is called as capsid, okay, or a protein coat. Okay, the protein coat is called as capsid. So let me write it here. So they have a protein coat which is called as capsid, and genetic material which is either DNA or RNA. So this is a very simple structure. Unlike eukaryotes or prokaryotes, which are quite complex in their organization, this is a very simple structure. And the genetic material can be DNA or RNA. Some of the viruses like herpes virus, hepatitis virus falls under the category of DNA viruses, whereas viruses like poliovirus, tobacco mosaic virus and rotavirus fall under the category of RNA viruses. Okay, let's move on. Okay, so let's look at how the viruses are classified based on their structural organization. So one of the key fundamental structure of viruses is that, as I said, they have a genetic material which is either DNA or RNA, and they are surrounded by a capsid which is a proteinaceous material. The capsid is not a single unit. The protein is not a single unit. The capsid is made up of several small units which are called as capsomeres. And the number of capsomeres in each of the virus is going to vary. Okay? And this is the genetic material which is surrounded by a protein coat. In addition to that, some of the viruses, they have an additional external coat like this, and this one is called as the envelope, okay, and which is primarily made up of a lipid or fatty acid derived material. Okay, so some of the viruses have an external coating, while other viruses do not have that external coating. So the viruses which have an external coating are called as enveloped virus, because they have an additional lipid layer around the capsid. Whereas the viruses like these, the examples are poliovirus, adenovirus and norovirus. They have just a very simple structure, the nucleic acid and the capsid. They do not have an external coating which is the lipid based layer. Okay, so let me check if there are any questions so far. What is the difference between DNA and RNA? Okay, this is a very simple question. So DNA is called as deoxyribose nucleic acid, whereas RNA is called as ribose nucleic acid. So as you know that nucleic acids are fundamentally made up of three different units. One is called as a sugar, then nitrogenous base and phosphate. So these are the three basic essential units of any nucleic acid or nucleotide. The sugar that is present in the nucleotides is a pentose sugar. So why it is called as a pentose sugar? Because it has 5 carbon atoms. So therefore this is called as a pentose sugar. So the pentose sugar which is present in RNA is called as ribose. Okay, ribose. Whereas the pentose sugar which is present in DNA is called as deoxyribose. As it is a deoxyribose, therefore this is called as DNA. So that is the fundamental difference between DNA and RNA. The sugar is different. In addition to that, DNA is usually single stranded, whereas DNA is usually double stranded, whereas RNA is single stranded. So this is also another key differences between DNA and RNA. So let's move on. Okay. So the classification of viruses are also done with respect to the host they infect. So if they infect plants, they would be called as plant viruses. One of the very key example of this group is tobacco mosaic virus. As you can see here, So the outside coat is made up of different capsomeres, and one of the questions that usually come in the exam, how many capsomeres this organism has. So this has 2130 capsomeres. Okay, so their entire capsid is made up of 2130 capsomeres. Okay, and this is one of the example of plant viruses, and their genetic material is RNA. Most of the plant viruses have their genetic material is RNA. Okay. The next group is the animal viruses. So they mostly infect animals, including human being. The other one which we are specifically going to discuss today is the bacterial viruses. These are the viruses which infect only the bacteria. So therefore they are called as bacteriophage. Why they are called as bacteriophage? Phage means eating. Okay, phage means eating. So as they infect the bacteria and destroy the bacterial cell, so therefore they are called as bacteriophage, or simply we call them as phage. Okay, so they were first studied by a group of scientists called as Stuart and D. Harrell. Okay, if you look at these two scientists were studying on bacteria, and once they found that one of the bacterial, suppose if you see the left one is a culture plate of bacteria, and the bacteria is growing happily over here. Okay, and on the right side, on this one, you see this bacterial culture, and you see a lot of plaques around here. Okay, the plaques are the spaces where the bacteria is not growing, because here the bacteria has been lysed completely. Lysed means they have been destroyed completely, and destroyed by what? So they destroyed by viruses. So when they identified why the bacteria is not growing at this specific region, they found that this specific region has some nucleoprotein materials, and when observed carefully, these were observed as some bacteriophages. And if we just look at the sheer number of the bacteriophages, those are present in the universe, they could range somewhere 10 to the range of 10 to the power 31 in terms of their number. The number is extremely large and diverse. Why they are diverse? Because they are infecting one of the largest kingdom in the world, which is bacteria. So as they are infecting bacteria, their number is extremely large. Okay, and it would be, if we combine all the life on this planet, including plants, animals and bacteria together, the bacteriophage would actually supersede all of them. So this is how a bacteriophage infects a bacteria. What it does, okay, this is a typical structure of bacteriophage which we are going to discuss. So the bacteriophage only inserts its genetic material into the bacterial cell. So if you see, this is a typical bacterial cell. Onto this bacterial cell, the bacteriophage comes and sits on the bacterial cell. Once it sits on the bacterial cell or attaches on the bacterial cell, it injects its genetic material. Once the genetic material is injected inside the bacterial cell, then the virus will start multiply inside the bacterial cell. Once they will start multiplying, they will produce large number of bacteriophages. And this is how the bacteriophages start growing inside the bacteria. So let us look at that process in a much more detail. Okay. So this is a typical structure of bacteriophage. So this has three parts essentially. One is the head, the other one is the tail, and the middle part is called as the collar or the neck. So the neck connects the head to that of the tail. The head consists of two fundamental units. One is the nucleic acid, and the nucleic acid is mostly either DNA or RNA, and it has a protein coat which consists of the capsids. And as you can see the shape of the head, it is mostly icosahedral. So icosahedral is a polyhedron consisting of 20 different faces. As you can see, it has multiple faces on the head. So this is a icosahedral in shape. and the head is connected to the neck, and the neck is connected with the tail. As you can see, the tail is covered with a sheath. Sheath means it's covered with a protein like covering, and the sheath is contractile in nature. Please remember this thing. This is very, very important. This is a contractile sheath. Contractile sheath means it has the ability to get contracted. That means it can contract as well as relax. So when it will contract, what will happen? The shape of the size of the virus will reduce because this is getting contracted. And when it is contracted, what will happen? The DNA which is present here will be injected into the bacterial cell due to the contraction of the sheath which is present in the tail. Okay, and that is the most essential feature, how only the DNA which is present in the head is going into the bacterial cell. It is because of the contraction of the sheath which is present in the tail. Okay, and also they have many tail fibers, and the tail fiber number varies depending on the type of bacteriophage. Some of the bacteriophage may not have, and some of the bacteriophage will have many number of tail fibers. And these tail fibers have essential proteins which can recognize the bacterial surface. As you know that, suppose this is a bacteria, and on this surface of the bacteria there are some specific proteins which are called as receptors. Okay, and these bacteriophages will go and specifically attach onto these receptors. Like for example, if you want to identify a place where you want to go, how do we identify a place? Because we have to know the exact location where it is located. So we put that location on Google Map and we go there. Similarly, so this is the post box. Okay, so these receptors as the post box for the bacteriophage. So they identify the location first and then attaches there. Okay, and what identifies that post box? These tail fibers. So these tail fibers have some specific proteins which can recognize the proteins present on the receptor of the cell wall of bacteria. And once they recognize there, they sit onto that cell wall, and the next process will begin from there. Okay, and there are some additional structures also present at the base of the tail, and these are called as spikes. And these spikes are usually 6 in number, and their number can also vary. Okay, and these spikes creates a hole, just like an injection syringe. It creates a hole. Similarly, these spike proteins, okay, are quite they can create a hole on the bacterial cell. As they can create a hole on the bacterial cell, and the contractile sheath can be contracted, and that is how the DNA can be injected onto the bacterial cell. Okay, so these are the different types of bacteriophages that we see generally. Okay, for you have to remember only few of the important bacteriophages. One of the important bacteriophages is 5X174, and what you need to remember in that bacteriophage, that this is a bacteriophage which is composed of single stranded DNA. As you know that DNA are usually double stranded, so this is an exception. In exam, usually most of the exceptions are asked in question because these exceptions are variabilities. Okay, so the 5X174 has single stranded DNA. Okay, then you have double stranded DNA viruses, which includes the lambda and the T even series. Okay, so these are the three things that you have to remember mostly for the with respect to your examination perspective. Okay, so this is the mode of reproduction. So let us look at what I was discussing earlier in brief. In detail, let us look at the overall process. So the first step is the adsorption of the viral particle or the phage particle. If you look at here, the phage is attaching itself onto the receptors. I said that the bacteria on its cell wall contains some receptors, and the tail fiber will specifically attach onto the receptors. Once it will attach onto the receptors, the contractile sheath which is present here on the tail, that will contract, and that will lead to the movement of the genetic material, which is DNA, which was present in the head, from the bacteriophage onto the bacterial cell. Okay, so now the DNA is injected onto the bacterial cell. So this is how the overall mechanism works. Okay, and once it goes inside the bacterial cell, the real activity of the bacteriophage begins there, because until this point of time the bacteriophage actually was not so much of active. Now it will start becoming active. Okay, what it does, the bacteriophage takes the control of the host cell machinery. So host का जितने cellular machineries है, for example its replication machineries, its protein synthetic machinery, everything will be taken control of by the bacteriophage, by the virus. So now virus is the boss and the bacteria is its servant. Okay, so now the virus will guide the bacteria what to do. The bacterial enzymes will function as per the guidance from the bacteriophage. So that is what exactly mentioned over here. The enzymes coded by the bacteriophage genome shuts down the bacterium's macromolecule synthesis, means the DNA synthesis and protein synthesis will be stopped. Okay, the bacteriophage now synthesize its own genome, because whatever was being utilized for the bacteria will be all routed for the utilization of the bacteriophage. Now they will start using for themselves. Okay, then synthesize bacteriophage structural components. So then as soon as it takes control of the host cell machinery, they will take control of, and they will start making its own proteins, own DNA and everything. And this is how the overall replication process works. Okay, so there are some questions, let us look at them. Okay, so one of the question is that outside the host cell viruses like dead. Sir, what is the difference between RNA and DNA? So this is what I have answered earlier. Sir, I have heard that bacteria are helpful to us. Is that true? Yes, all the living organisms, including the viruses, they are helpful to us. Okay, for example, if the bacteriophage would not have been there, what would have happened? The bacterial population would have exploded like anything. Okay, and that is dangerous for us. Okay, so these are the natural organisms which are, these are like natural antibiotics, which are we are going to talk later on. Okay, because they are helping to control the rate of proliferation of bacteria. That is why they are useful to us. Similarly, bacteria are also useful to bacillus, which is helpful in the production of yogurt. So therefore, bacteria, some of the bacteria are useful to us. For example, you know, most of our stomach, our intestine, okay, in that matter, is filled with a large number of bacteria. These are useful bacteria. Our skin has a large number of microflora. Why our skin has a large number of bacteria? Because that are good bacteria. Okay, and these good. Is there any other question? What is the meaning of peplomer? Peplomer is something I have not heard about. What is peplomer? Is it polymer or peplomer? Can you just clarify your question? Okay, so the next one is, after the virus starts to replicate inside the host cell, now it will start to make its own components, because it has started to replicate, it has started to make its own proteins. Now it will start to make it assembly, because virus is made up of two simple things. One is the DNA and the proteins. So now the DNA and proteins will be assembled, and now new viral particles would be generated inside the host cell. Okay, so many viral particles are generated inside the host cell. Okay, once these so many viral particles are generated inside the host cell, now the nutrients present inside the bacteria is not enough to support them. So what will they do? So they will run away. So how will they run away? So they will cleave the bacterial cell. How will they cleave? So the cleavage always happens with the use of some enzymes. Okay, they cannot cleave as it is. Okay, so they have to break the cell wall with some enzymes. and there is a specific enzyme, okay, which can cleave the bacterial cell. As you know that the bacterial cell is made up of a specific chemical component called as peptidoglycan. So this is usually comes in the examination, okay. What is the chemical composition of the cell wall of bacteria, which is peptidoglycan. So as you can just see this word, peptido. Peptido means something related to peptide, right? And glycan is related to sugar. So peptidoglycan is made up of sugar and peptides, and the enzyme which the bacteriophage produced to cleave this particular peptidoglycan is called as lysozyme. So lysozyme is the enzyme which breaks the cell wall, okay, and lysozyme is also present in our tears. Why it is present in our tears? Because tears, eyes are one of the opening to bacteria to get inside our body. It's one of the route through which bacteria can go. So therefore tears are produced to kill the bacteria. So tears usually contain a large amount of lysozyme, okay. So once lysozymes are produced, there would be cleavage in the bacterial cell wall, okay. So the bacterial cell wall will break. So once the bacterial cell wall will start breaking, what will happen? The bacteria leaves under high, the inside the bacterial cell there is cytoplasm, okay. The inside the bacterial cell we have the cytoplasm, and the cytoplasm usually contains a high amount of salt and sugar and other components. So therefore the water potential, okay, inside the bacterial cell is low. Because how the water potential is, because you know water potential of pure water is the highest. As some components would be solubilized in the water potential will decrease. But the outer membrane of the outer layer of the bacteria is the extracellular layer, right? This is the extracellular space, and the extracellular space contains water. So the water potential here is always higher, and always water moves from the higher water potential to the lower water potential. So what will happen now? So water will move from here to here. So as water will move from outer space to the bacterial cell will now burst completely. So as the bacterial cell will burst, the bacteriophage which are present inside the bacterial cell will come out and infect a new bacteria. So that is what is the next step. Hope this process is clear, right? So once they are released from due to the breakage of the bacterial cell, so they will grow and reinfect another bacteria, okay. So the release is accomplished by two things. Please remember this thing. This is usually comes in the exam: lysozyme, action of lysozyme, and complete breakage of the cell wall due to osmotic lysis. Whatever I discussed, okay, that the water potential of the bacterial cell is lower, okay. So water moves from higher water potential to lower water potential, and therefore completely lyse the bacterial cell, okay. So this is the fundamental mechanism how the bacterial cell wall breaks, okay. And usually during one infection cycle, okay, around 50 to 100 phage particles, phage particle means new bacteriophages, are produced from one bacteria, okay. So it's a huge number. From one bacteria, 50 to 100 more new bacteriophages are produced in one cycle. So the time it requires from the adsorption, okay, when the bacteriophage adsorbed onto the bacterial cell wall till their release. So when it started infecting till it is started producing, and now it started producing. So the timeline, okay, so gap, okay. So if you look at, so entry to production and exit, right? So this is the timeline, okay. And this time gap is called as a eclipse period. Eclipse period. So eclipse period is the time required by the bacteriophage to enter inside the bacterial cell and then reproduce inside it and then come out, okay. So that is usually called as the eclipse period, okay. So let us look at the different phases, that the different ways in which the bacteriophage infect a bacteria. One of the process is called as a lytic phase, and the other one, which is most interesting one, is the lysogenic phase. The lytic phase is called as the virulent phase. Why it is called as virulent? Because lytic means lysis, because it is immediately killing the bacteria. So that is why it is called as the lytic phase. Lysogenic phase is called as temperate phase. That means in this phase the bacteria is not killed instantly. Okay, so that is why it is a mild stage, okay, or a temperate stage. This kind of stage is usually seen during the infection of T phages, T type of bacteriophages in E. coli. And this is seen when a lambda type of bacteriophage will infect E. coli. Okay. Is there any more questions? Sir, what is the size of bacteria? An average bacteria will be 1 to 5 micron in size. I hope you know what is 1 micron. It is 10 to the power minus 6 meter. Suppose if you take 1 meter and divided by that by 1 million, a fraction of that is going to be 1 micron, and that is so small to be visualized through our naked eyes. So therefore usually we see the bacteria in microscopes, whereas the viruses, their size is even smaller. They could be somewhere between 100 nanometer, around that 100 to 500 nanometer. Okay, so the size is even smaller, much smaller than the bacteria. Okay. Okay, so let's move on. Okay, so this is what is the Lytic cycle. So in the Lytic cycle, as I said, that it's a virulent phase. What happens here? So the bacteriophage attaches onto the cell wall. Okay, we'll quickly move because we have discussed most part of it earlier. Okay, it will inject its DNA into the bacterial cell. As you can see, this green color is it represents the DNA. And as soon as the DNA will get inside the bacterial cell, it will start to multiply. Okay, it will take control over the host cell machinery. It will make more copies of DNA, and also it will make its own protein. Protein means the capsids and the tail fibers, everything. And then the complete bacteriophage will be produced, and then they will lyse the cell wall. As they lyse the cell wall instantly after they enter into the bacterial cell, that is why it is called as Lytic cycle, because they don't wait for some time. They instantly kill the bacteria as soon as they go inside. They multiply immediately. Okay, they don't wait for some time for multiplication. That is why this kind of cycle is called as Lytic cycle. Whereas the lysogenic cycle, which is the most interesting phase, okay, as you can see here, once the DNA enters into the bacterial cell, it will not start to produce many copies of the viral DNA immediately. What it is doing, as you can see here, the DNA of, this is the DNA of the virus. The DNA of the virus is integrating into the genome of the bacteria. Into the genome means into the DNA of the bacteria. It is not producing its own DNA. It is integrated into the DNA of the bacteria, which is the most differentiating feature from that of the Lytic cycle to that of the lysogenic cycle. Lytic cycle में क्या हुआ था? The bacteria immediately started producing its own DNA and then proteins and created more phage particles. But here it is not doing that. It is integrating inside the bacterial DNA and not causing any harm to the bacteria. It is only integrating into the bacterial DNA. Okay, and also as the bacteria divide, so they also kind of replicate, because as the bacterial DNA will divide, multiply, they will also multiply. But when a unfavorable condition comes in, the DNA of the bacteriophage recognizes that I am in danger, okay, I am in stress. So what it does, it gets excised. Excised means it gets removed, as you can see here. Now it is removed from the bacterial DNA. And as soon as it gets removed from the bacterial DNA, then it will start the Lytic cycle. Okay. So what it will do? It will start doing the same way the Lytic cycle began. Okay. And why the lysogenic cycle converted to a Lytic cycle? Because of the application of the stress. And in exam, it can be asked, what are the stressors? The stressors could be UV, one of the most important stressor, okay, because if UV light is given or irradiated onto the bacteria, so then it acts as a stressor, so that the chromosome is excised from the bacterial genome. Okay, there are some further questions. Let us see. About 1 to 2 micron in diameter and 5 to 10 micron in long. Sir, what is fungus? Okay. So fungus is a separate class of organisms. Okay, if you know the 5 kingdom system of classification, which was proposed by Whitaker. Okay, let me add another set. Okay, so in terms of the 5 kingdom system of classification, the entire living organisms are classified into 5 kingdoms. The first kingdom is the Monera, then Protista, then fungi, then Plantae, and then Animalia. Okay. So Monera includes all the bacteria. So bacteria means they are all the prokaryotic organisms. Prokaryotes. So Monera includes all the prokaryotes. And the rest others are all the eukaryotes. Then how all those eukaryotes are placed on separate kingdoms? Protista, for example, it is a single cellular eukaryote. Please remember this thing. This is a single cellular eukaryote. Only one cell. For example, amoeba, paramecium. These are all single cellular eukaryotes. So therefore, they come under the kingdom Protista. Whereas fungi is all these 3 are multicellular organisms. Okay. What is the difference between then fungi, Plantae and Animalia? Fungi is a multicellular organism which cannot make its own food. They are heterotrophic. That means they cannot produce its own food. That is why they are placed in a separate class or kingdom altogether. Plantae, they can make their own food. Okay, so they can make their own food, so they are autotrophic. What are animals? Animals are heterotrophic, but they have a different mode of nutrition, which is ingestive mode of nutrition. So that is why they are placed on a separate kingdom. So if you have to define what are fungus, so fungus are a group of microorganisms which are multicellular and heterotrophic and have heterotrophic mode of nutrition. Okay, I hope there are no further questions. Let's move on. Okay. Okay. So when the phage DNA integrates into the bacterial DNA, the integration is not a very simple step process. It requires something. How it has to be integrated? It requires the help of an enzyme to be integrated into the bacterial genome. And what is that name of the enzyme? It is called as integrase. So integrase is an enzyme that helps the phage DNA to be at this particular phase. It is inert. Bacteria occurs in 3 mechanisms. One is transformation, then the second is transduction, Then the third mechanism is conjugation. Okay. If you look at transformations, for example, what in case of bacteria, the sexual reproduction is not a real sexual reproduction. Okay, it is just the transfer of gene. But how the transfer of gene occurs? In transformation process, the gene is directly taken up by the bacterial cell. Suppose this is a bacterial cell and this is a DNA outside. The DNA will be directly integrated into the bacterial cell. So that is called as transformation. So transformation is a direct uptake. Transduction is a process in which the gene from one bacteria or the DNA from one bacteria is transferred into the another bacteria with the help of a bacteriophage. So that means, suppose this is one bacteria, it has its own genome, this is another bacteria, it has its own genome, say this is the genome. Suppose this genome of this bacteria is integrated here by some way. And what is the mode in which it is transferred? It is transferred with the help of bacteriophage. Then such kind of mechanism is called as transduction. And what is conjugation? Conjugation is a process. For example, this is a bacterial cell, this is another bacterial cell. Okay, so there is a DNA here and there is a conjugation tube that would be formed. The DNA is going to be replicated and this replicated DNA will be transferred here. So this is a this is a transfer between from one bacteria to the other bacteria with the help of a conjugation tube. Conjugation tube. So this is called as conjugation. So there are three modes of reproduction in bacteria. So we are going to discuss how the bacteriophage helps to transfer DNA from one bacteria to the other bacteria. Okay. So for example, as I told you that during the lysogenic cycle, what happens? As soon as the phage DNA comes inside, it integrates into the bacterial genome. When it is integrating into the bacterial genome, suppose there is a stress coming up. And during that stress, what happens? I told you that the viral DNA is going to be excised. When it is getting excised, see, look at here, what happened? It is getting excised from the bacterial genome, but it is taking some genes of the bacteria also when it is excised. If you see here, some portion of blue color is marked here. Okay, so that means when it is getting excised, not only it takes the viral DNA out of that, it also takes a bacterial DNA along with it. So now it will replicate and produce multiple copies, but the gene which is present inside the bacteriophage now, it is not the gene of the bacteriophage alone, it also has the gene of the bacteria. It has both the genes. Now suppose this bacteria go and infect, this virus go and infect another bacteria, what it will do? It will not only transfer its DNA to the bacterial DNA, also it will transfer the bacterial DNA, this one also. So now the DNA of one bacteria, suppose this is a bacteria A and this is bacteria B. So if you see here, the DNA of bacteria A is now going to be transferred to bacteria B. And what helped in that process? The bacteriophage helped in that process. So that is why this process is called as transduction. And why this process is so significant? We know that there are some bacteria which are resistant to antibiotics. If, for example, these viruses will transfer that antibiotic resistant gene of one bacteria to another bacteria, what will happen? The other bacteria will also become antibiotic resistant. So this is how most of the bacterial species develop antibiotic resistance. So that is why it is so much relevant to us. Okay. One of the important discoveries in molecular biology was done using the bacteriophages, and it is the discovery of DNA as the genetic material. Okay, because until 1950s, early 1950s, it was believed that the protein is the genetic material. Why? Because the protein are made up of amino acids and there are 20 different amino acids, whereas the DNA is made up of only 4 different nucleotides. And if 4 different nucleotides will be recombined with each other, the number of combinations they can make would be very little. Whereas if there are 20 amino acids, the number of combinations it will make, it will be very huge. So that is why people used to feel that protein is the genetic material. But it was Martha and Chase, okay. So Hersey and Chase, sorry, not Martha and Chase, Hersey and Chase. For the first time, they proved that DNA is the genetic material. So for proving that, they have taken bacteriophage as the model organism. Why bacteriophage, not any other organism? Because if we take any other organism, that is very, very complex. For example, for a bacteria, or it contains proteins, sugars, lipids and so many other components. Whereas what is the component of a bacteria? It has only protein and what else? It has only DNA. Only 2 things. Okay. So to prove that either DNA is the genetic material or protein is the genetic material, we have to only consider or label these 2 components. So what they did a very simple experiment. They radio labeled the protein component and separately, and they radio labeled the DNA component. How can you radio label separately DNA and protein? Because you know that I told you just before some time that DNA contains phosphate, but protein do not contain phosphate. So that means DNA has phosphorus group, whereas protein contains sulfur and DNA doesn't have sulfur. So they radio labeled specifically the protein using a radio labeled sulfur and the radio labeled DNA using a radio labeled radioisotope of phosphate. So now for example here in the first experiment, experiment 1, they have only coated with radio labeled sulfur, and if radio labeled sulfur would be used, what would be radio labeled? Only the capsid part, which include the head, the neck, the tail and the tail fibers, not the DNA, and then they infected the bacteria. And what they found? That the bacteria is not showing any radioactivity. So if the bacteria is not showing the radioactivity, that means the protein component is not going inside that. In the next experiment, experiment 2, they have radio labeled specifically the phosphorus, and the phosphorus will radio label means the DNA will be radio labeled, right? Clear? So if the DNA is going to be radio labeled, so now when they check for radioactivity, they found that the bacteria is showing radioactivity. So bacteria is showing radioactivity. What does it confirm? That means the DNA is going inside. So because of this discovery, Delbruck, Hersey and Salvador were awarded Nobel Prize in 1962. Okay, because for the first time they demonstrated the fact that DNA is the genetic material, and this experiment clearly proved that. Okay. So now we'll take up some questions. Okay, so let us look at the first question. Transfer of genetic material from one bacterium to another in the process of transduction is accomplished by. I ask all of you if you can post your answers in the chat box. Okay, I told you what is transformation, what is transduction and what is conjugation. So the question that is asked, in the transduction, the transfer of genetic material is done by what? Okay, any questions? No questions so far? We'll wait for a minute. Okay, so as I told you that transduction is a process of transfer of gene with the help of bacteriophage. Let us look at the Options: conjugation between bacteria of opposite strain. Conjugation is a different process. It is not transduction. So this answer is incorrect. Physical contact between donor and recipient. No, this is also incorrect. Direct uptake of DNA. Direct uptake of DNA is transformation. So this is also incorrect. With the help of bacteriophage, I said that with the help of bacteriophage, it's transduction. So that is why this is going to be the correct answer. Let's look at the next question. When the viral genome gets inserted into bacteria host, it is termed as replication, Lytic cycle, Lysogenic cycle, and capsid formation. So it is saying that if it is getting inserted into the bacterial genome, okay, it's not bacterial host, it's bacterial genome. So if it is getting inserted into the bacterial genome, it is termed as what? Lytic cycle. Okay, Lytic cycle is not the process in which it is getting inserted inside the bacterial genome. Okay, it is the lysogeny. So lysogeny is a process in which it is getting inserted into the bacterial genome. Let's look at the third one. Okay, so this is very easy. So identify the different components which are labeled in A, B, C, D. Okay, so as I told you, the bacteriophage has essentially 3 components. You will have to label them. Okay, just 1 minute I will wait and then I will answer that. Okay, someone is saying C. Okay, why C? So let's look at whether it is C or not. So A is head, correct? A is head. Then B is collar, yes, B is collar. C is sheath, yes, C is sheath, okay, contractile sheath that we have discussed, and D is the tail fibers. Yes, C is absolutely the correct option for this one. Okay, let's look at the other question. Conversion of lysogenic to lytic cycle occurs in, okay, there are certain conditions given here: favorable condition and high nutrient availability, exposure to stressful condition, exposure to UV light, both B and C. Okay, Hardik is saying it is D, both B and C. Yes, absolutely, it is both B and C. So under stressful conditions including UV, okay, the lysogenic cycle will be converted into a lytic cycle. Okay, so this is what we had for today's discussion. Please feel free to post your questions, any clarification that you want to have after this session. Also, what actually you would like us to discuss in the next round of discussion, please post your views around that. Thank you very much.